it never took off, despite being a much more highly effective keyboard. I've been interested in what drives innovation, and how do we make progress, and how do we change clinical practice. Scientific research is launched with major breakthroughs or reconceptualizations that provide model problems and solutions. And recognition of the new paradigm sets off an intellectual frenzy as scientists and clinicians are attracted to the field either to advance or disprove the new conceptualization. And then scientific consensus about the field then is developed. The journey begins with an idea. It proceeds with the research, and the journey then goes through several steps before there's adoption in clinical practice. I have two recommendations for those of you who are engaged in the journey. One of them is to hook yourself up to mentors, people who are smarter than you, people who have more wisdom, and people who will work with you. And I was very fortunate, of course, to make a connection with Paul Kaufman, our guest of honor 25 years ago. And Paul and I have collaborated on the outflow pathway for almost 25 years now. That's a subject that I discussed 10 years ago in the inaugural Clinician Scientist Lecture. The other recommendation is to surround yourself also where you live with people who are smarter than you, who are collegial, who are collaborative, people who you can sit around the round table with, who can give you advice. And I've been blessed to have a group of people who've been able to give me advice, beginning first with Pam Sample. Rigby Slight has been a great source of advice to me over the past 25 years. Linda Zangwill, John Liu, Chris Bowd, Mike Goldbaum, Felipe Medeiros, Jamie Lindsay, and most recently, Kang Zhang, a brilliant geneticist who's working with us now to teach us about the application of human genetics to glaucoma and stem cell biology. So let me tell you about some of 10 changes that I think that might move our field forward. And these are some questions that need to be answered before we can really move forward. And the first is a very simple question. What is glaucoma? We've been listening yesterday and today to our colleagues discuss their interesting and significant research. But how many of us really understand what is glaucoma? Well, if I asked you what is glaucoma, many of you would say it's a characteristic optic nerve disease with those well-known visual function changes and changes in the optic nerve. Glaucoma is a continuum from undetectable disease to asymptomatic disease, and some patients proceed through the continuum and become functionally impaired and even blind. Most definitions of glaucoma talk about structural and functional changes. I think we're on the verge of adopting new definitions of glaucoma, ones that'll be more specific and more sensitive. With some of the new instrumentation that we have for performing structural and functional tests, I think we have the possibility to have a more specific diagnosis for glaucoma that relies upon progressive structural or functional change, and not simply a static comparison of the optic nerve structure and function. There are other ways to diagnose glaucoma as well. There are biologic definitions in which we go into the laboratory and we increase intraocular pressure and create changes in retinal ganglion cells. And there are epidemiologic definitions of established visually significant organ damage. Perhaps the most exciting change that we might see in the relatively near future is a definition that's both specific and sensitive and based on the genetics. And I'll say something a bit more about this very shortly, because the second item I wanted to bring to your attention is our ability to develop and refine predictive models to better determine who benefits most from treatment and who does not. What do we do about the patient who comes in and asks us, when they learn they have glaucoma, will they become blind? Will they become visually impaired, and will their quality of life be impacted? Several years ago, with my colleague Felipe Medeiros, we developed a very primitive and crude predictive tool based on OATS data and based on data from the Diagnostics Innovations in Glaucoma Study, or DIGS, that we have at the Hamilton Glaucoma Center at UCSD with Pam Sample and Linda Zangwill. And more recently, predictive models have been refined. And you can go onto the web now and find that there are much better predictive models than we even envisioned a few years ago that have been established based on larger databases. However, these predictive models, also known as risk calculators, can undergo evolution. And perhaps the evolution and the change that will bring the most to predictive models is something that introduces genetic component to the predictive model. This is data that comes from the Kang Zhang laboratory, in which a new gene for glaucoma is identified. And it's one of what I expect will be many new genes for glaucoma as large databases are developed. Janie Wiggs is leading the NEIGHBOR study, in which up to 2,000 samples of blood are obtained from patients with glaucoma. And it's likely that a database like this will generate new genetic loci and targets for diagnosis and treatment. In this particular one, we have the association of SNPs in chromosome 2 with primary open-angle glaucoma. And if you look at the minus log p values from the association analyses on top, you see that there are some just extraordinarily likely genetic focuses for glaucoma. Another question, what damages the optic nerve? Well, this continues to be debated, debate that began more than 100 years ago. And throughout the meeting, we're going to hear over and over again, glaucoma is a disease of pressure. It's a disease related to the blood flow. It's a disease related to ischemia or something else. Here's that summary slide that I showed earlier today that talks about the multiplicity of potential damaging factors for the ganglion cell and for the axon. Well, how are we going to determine what damages the optic nerve in glaucoma? Well, we need some easy-to-use models. We've been plagued by the absence of a model that truly mimics human open-angle glaucoma or human closed-angle glaucoma. We have